Any problem? I think there is some confusion. This lecture is later on. Can you stop the link little? Pause the link. No, no, ये मेरा lecture है. Confused क्योंकि tutorial. Sorry, just pause the link and then we can restart. Okay. So blast and all those tools we will take up because that is connected to tutorial. So it will be difficult for you to cope up. So this I am taking lecture 8 one. Okay. Then 6 we will be taking later on. So this lecture 8 overview of system biology tools and gene network finding. This is the lecture 8, and if you see, since we have strength in microbial systems biology, we are microbiology learners, so we focus this lecture according to that, but this can be used in other fields as well. So this aspect of microbial systems biology cannot be learned without learning the gene network within the microbial systems. So we are taking example of microbial system, but you can also think about various other systems which you want to use. And what we are trying to learn over the years through gene network is that the engineering of the PGPMO metabolism, especially the people who are working in plant growth promoting microorganisms, various microorganisms which are giving good benefits to the soil and giving good benefits for the crop. So this is one area which is totally neglected, especially with the help of gene network. You can see most of the aspect. People are focusing on biofertilizers, other aspects, okay, making crops more resilient towards the drought and all other things. But the part of how these microbes are affecting, like you people might be saying, how these microbes are affecting and what is happening in the rhizospheric community and how these microbes are triggering the response for better resilient crop, that has not been studied in network level. So that is the area where you can think, and we will be focusing on that. One is functional genomics, you know about it. So functional genomics is also one important area which has lot of potential in personalized medicine as well as on other aspects of understanding microbial community. The third one is the metabolic models, how the metabolic models can be generated. So if you see, the tools which you can use is given in this table, and this table I prepared in 2014. Still we are not able to use all the tools in last 10 years. So these tools are very good tools, and I don't know how many of you are using these tools, but all aspects, whatever analysis you want to do, you can do using these tools. So if you want to use the tools for metagenomic data analysis or any type of data analysis, you can definitely use these tools. Like for functional category, if you want to know about the functional category, about the KEGG orthology, then NOG, you can use MEGAN, MEGAREP, MGRAST. So MGRAST tool, people are using it. Taxonomic analysis, if you want to know specifically about the taxonomy of the microbes or any organism, you can use these tools, QIM, MOTHUR and MLST. These tools can be used, and they can tell you about the domain, phylum, class, order, family, genus, all details you can get. Clustering, If you want to understand the clustering, how various diversity index you want to know, so you can use these tools of OUT, table, tree building, alpha diversity, beta diversity, taxonomic composition, rank abundance, and the tools are Galaxy, IMG, GM, and Megan. These tools are very means readily available. Many people are using it. If you see any paper related to metagenomic data analysis or metabolomics, you will see the use of these tools. Then for pathways, this is a very new area, pathway analysis. Many people, they are trying to see the pathway, especially when they target for bioremediation, they target for something related to the disease, which pathway is connected to this disease, okay, and if they are connected to certain disease and certain aberrations, so the pathway will give very good clues. So for pathway analysis, you can use KEGG, SEAT, and METASET. So these tools can be done, that camera, MetaLook, and Unifrac. So if you are using these tools in your research, you are advancing your data to some next level, and also you are getting some more data to understand future potential of that particular data. So when you talk about omics data analysis, these tools, all tools you cannot use in one study, but at least one or two tools if you use, that will be better, and that will shape up your research in a better way. So this so if you see this one, nowadays concept is little complicated, and you cannot use one technique to learn your field. You have to have the, like we talk about multi-omics, okay. So when we talk about multi-omics and biotechnology or various type of tools, we have to use the multi-level technologies. So this is not possible with the help of one person or with one knowledge. You need to understand other fields as well, and as I told you, you need to collaborate, you need to learn from each other. So this is one model which I adapted from Otero and Nelson, and there is something which industrial systems biology or industrial biology, when we talk about the systems biology, how it is connected to industry, how it is connected to application, how it is connected to other fields, that you have to see. So I have given one example, that if you have one production host like yeast cell is there, okay, and you want to know more about that yeast cell, okay. People normally they think that yeast Saccharomyces cerevisiae is used for alcohol production or other type of beverages, but there are lot of things in yeast model. So I have given one example of yeast cell. If you want to understand that yeast cell, what are various techniques you can use. There can be a plant cell also in place of yeast, there can be animal cell also in place of yeast, but in yeast cell, how many aspects you can cover using various tools and technique, and that gives the clue for industrial systems biology. It is not system biology, industrial systems biology, where you try to see yeast, its genome, its analysis using data integration and models generation. Then if you understand the model of that particular metabolism, then you try to see its physiology, you see its transcriptome, see metabolome, you see flux analysis. If you are able to get that, you will able to know that, okay, your yeast species is giving the optimal production, or if it is not giving optimal, you want to increase the production of any type of metabolite, so you can do the strain modification, strain engineering. So strain engineering, for that you also understand that what is metabolome, what are the specific genes, what genes you need to target to get more production of yeast. So you do the modification of the strain, you do all these aspects with the help of your previous data which is available. If you take as a yeast as a wild strain and a modified strain, you can know the difference. Then only studying this is not enough. You need to understand that how growth analysis or growth profile of yeast is behaving. What are different aspects covering? So you do the various type of modeling. You see what fluxes are there which are controlling the production of yeast, and then Sorry, I. So yesterday we were talking about mobile and distractions. You see the distractions. Okay. So you do different models. Try to see what enzyme, what product you want to get through that. So these models will give you idea about that. Okay, the production level at 48 hours, 50 hours, or 55 hours, or in a certain scale. Then this is not only the way. You can also build kinetic models out of it. You can also see that the production kinetics, growth kinetics, so you built up kinetic models. So when we were working with cyanobacteria, we saw various other models also. So you can also think about the kinetic models of the growth profile. Any microbe, if you are studying, and if you are not studying its growth profile, any microorganism, any cell, okay, or any type of organism you are working, if you have not seen its growth profile, your studies are limited, because you don't know what is the growth kinetics. So growth kinetics is very important, and then you get all the information together, then you are able to understand each cell as a whole. If you want to study a cell, all these studies if you can do, you can understand the cells in a better way. I cannot say it will be the complete study of the cell, because there are so many genes, there are so many pathways, there are so many kinetic parameters, there are so many gene annotations, so there are so many things. But at least if you connect all this and you connect all these disciplines together and you learn together, you can have a better story from your learning. Otherwise you will study one part, another part will be left. If you study another part, some another part will be left. So at least you can cover one or two aspects in your research project, and based on this you can plan your projects also. So this is also idea giving you the project planning, because sometimes people are telling you apply for a project, you write a project, you give a project to a student, and a student is also coming to the supervisor and telling, okay sir, tell me what should I do. So this gives you a planning that okay, you can take up 2 parts of it, you can take 3 parts of it. It becomes a small project for you to learn in that specific manner. Going to further, if you see, there are some new tools which are used in understanding the PGPMO when we talk about the plant growth promoting organisms. So metagenomics, metatranscriptomics, and metabolomics, these are the new tools, and how they are important you can see, because there are few people who are working on plant microbe interaction also in this group. So they know about that these tools, they identify specific novel genes, and also they see that how transgenic plant can be developed using these specific tools, and designer plants can be also done. So when you use these tools and the soil microbiome is also linked together, you can also understand that only plant is not doing the job. It is the microbe which is giving the signals and trying to adapt accordingly, and the plant is also taking lot of benefit out of microbe. So this is a interaction thing, and the combination of new molecular techniques and genomics can give rhizosphere interaction or plant microbe interactions in the rhizosphere. I will show you another slide. Before that, there are 2 questions here to us, that why the soil microbial population is giving a specific signal for the growth of the plant. How these microbes present in the soil are connected to plant. When we talk about plant microbe interaction, why? We have to think about why we are talking about it, and also how it is happening. So these are the 2 things which need lot of questions. These are not the answers, but I will try to provide some answers. So the question is that a competition driven model for rhizospheric microbiome and a predictive framework of microbiome, how they can help us in better phytoremediation or better plant adaptations. So if you want to use microbe only for bioremediation, if you want to use plant only for bioremediation, it is not going to solve the problem. But if you use microbe based model, microbiome based model, or microbiome engineering based model, that will be better. So it is not possible without understanding the data involved here. So you need to understand the attributes which are making that particular plant to adapt to the new environment. So if you see, if you manipulate the plant, you do the genetic engineering in the plant, you do designer plant strategy, that is also not going to solve this problem, because you cannot control the microbial population within that vicinity. So this is a very important thing, and also this type of attributes are possible with the help of PGPMO engineering, that is the plant growth promoting organism microbiome engineering. So the approach can be the approach which we use in our lab, and you can also use this approach, is very common. We published this in 2018, that is the design, build, test, learn cycle. This cycle is very effectual cycle, people are using it now. Okay, when we published, the editor of the journal was asking, what is this cycle which you have made? It was a hypothetical cycle, and we made this cycle which is very helpful. Design, build, test, learn cycle. What it does, I will show you the cycle also. So what it does is that it identifies the effect of plant inoculation with these microbes, and also associated phenotypic changes which design the changes in the DBTL cycle. So once you see the cycle, you will be able to know. So this is a holistic approach, combinatory approach, which combines omics, transcriptomics, proteomics and metabolomics, and this is all data driven approach. This is all data algorithm driven approach. So if you see, I will show you this cycle first, then okay. So you can see this cycle in one of our paper which was published in 2018 in Trends in Biotech, and this is trends, this design, build, test, learn cycle. It highlighted the integration of systems biology and gene editing to improve the phytoremediation. So this is one example. I am not telling that all examples we can discuss it here. This is one example how this using the integration of system biology and gene editing can give you a better outcome. So if you see, I will explain little bit, and more terminologies, more aspects I will be giving you as a tutorial material. Yesterday also some tutorial material were suggested. Gaurav, please note it down, otherwise we will forget the papers, the tutorial material which need to be given to them. Okay. So this cycle, let me explain you. If you see this part, transcriptomic, if you see this network part, and if you see this improved phytoremediation, so this is complete cycle. If you want to study a microbe here, you try to see the transcriptome out of it, what is happening, and then once the PGPMO is inoculated, you see transcriptional changes at various level, like day 1, day 2, day 3, whatever, and you know the changes in the plant is happening when you inoculate the microbe, and you see that the plant at giving very good output or maybe not giving the output, you know everything. But how it is happening, what are the transcriptional changes, what are the transcriptomic changes happening, you can understand that. So same way, you use the tools of transcriptomics, you use metabolomics, you use the tools of proteomics, all together combined to understand that what network can be adopted for improved remediation. Like if the remediation has to be done for some heavy metals, or it is done for microplastics, or it is to be done for any other hazardous chemicals. So what network is involved, what pathway is involved for that particular chemical. So you identify out of it and try to see which network is playing major role, and then you build that network, which I will be showing in the coming slides. And then further, after getting the network identified, you also understand that okay, this gene is playing the important role there, and if you want to use the gene editing technologies, you can definitely think about gene editing technologies to target that particular gene, try to edit those sections, try to understand those aspects, and use the tools of gene editing for CRISPR-Cas9 to make your designer plant strategies and designer microbe as well. So both things are there, but only when we are using the design, build, test and learn cycle all together. So this cycle, these techniques, then plant microbe meta-organism. This is a new approach. You might have heard about metagenomics, you might have heard of organism, you have heard of the microorganism. I talk about meta-organism. So meta-organism is plant and microbe together. Okay, both together they are meta-organism. One plant is the organism, or plant microbe interaction is based on that. So you talk about this meta-organism approach, and this is a combinatory omics approach where the idea of meta-organism is successful implementation of omics strategies which provides clear understanding of the process involved in the decontamination process, and it can detect the signals which are not detected if they are studied individually. So if you study individual microbial signals there, you cannot get. It is the same story like people who are working on animal systems. Okay, you have lot of organs which are connected to various type of diseases. Okay, but you identify one disease and another disease is also existing in the body, and another disease is also there. So in the same way, if you study individual systems, you cannot do that. But if you are connecting every disease to each other and try to see that why disease is happening and what are the specific type of signals which are coming. So in the same way here, if you study the microbe separate, you study the plant separate, you will not be able to get these signals. You have to see the meta-organism. You have to see how these are connected. And the challenges which we face is that how the plants are triggering the assembly of beneficial microbes. Why plant is taking up the, what are the signals coming to take only beneficial microbes? Okay, like there is a question that why beneficial microbes plant is, what are the signals coming? Why they are not targeting or not taking up other microbes? So why, and what is the connection between that, and then how the PGPMO respond to stress signal? So when there is a stress like heat stress, okay, and you see the dot, also the water, okay. So what if there is a scarcity of water, if there is excessive heat, so how these signals are, and cold also, okay. So how they are able to survive. So this I have already explained. This is a tutorial material, and this is an excellent opinion tutorial material where you will read all that, and this will be very helpful those who are working on sustainable agriculture, and they are targeting plant growth promoting microbes, they are targeting bioremediation as well, environmental people, and also the persons who are looking metagenomics data analysis, okay, or metabolomics tools they want to use, okay. So this article is available with us, and Gaurav noted down to give them, and these articles are written in very simple language. So definition is there, and all specific things are there, and also the terminologies, how the terminologies could be used are given there. And this I collaborated with Supratim and Royal C. Rabra and Sangeeta Negi. These three were in US, still in US, all three, and they are leading one important group in New Mexico consortium, and now it is a national lab in US. So after this work they have started antenna engineering, so we focus that. Still there is not much work done in this idea, the idea is still available. So if anybody wants to use this idea in her research, they can use it. So future scope and application of this type of technology, what will be the applications? It will employ the knowledge of systems biology with gene editing tools, and it establishes the phytoremediation attributes, and if we are able to do successful implementation, which is very tough, it is not easy. So when we published this article, we got some emails from US, some companies which are looking for phytoremediation. So they discussed this aspect, that how we do in the large environment, how it is possible to do in the larger environment. So this is another question, and also the question of the diversity of the organism. If you see our last point, there are lot of organisms, lot of microorganisms, you know that. We know only 95, we know only 5%, we don't know 95%. So if we know only 5%, so there are so many microbes in the soil, and we are targeting that we want to understand their interaction, it is not possible. So the diversity of microorganism is another challenge, and their genetic diversity. If we try to focus one organism, two organism, three organism or consortia, it is not going to solve. Recently we faced this problem of diversity of organism in the rhizospheric soil. So if you read our recent paper which we published in M Spectrum research paper, there you will see that we use only one organism, because we started with consortia. We made a consortia, very good consortia mixed with fungal, bacterial all together, but we were not able to understand its function. So we thought, okay, we will make another consortia and then do the phase wise study. So we gone through that one organism and we got a better outcome out of that. But we may had a better outcome when we have a consortium. Okay, so as people say that unity is power. Okay, so in the same way, the microbial consortia, microbial organism, microbial combination, and microbial plant meta-organism is also a new aspect. So it needs a lot of research and exploration, which we can do in future. Another thing is that bioremediation with microbes is eco-friendly. So you can use this microbes for various type of bioremediation attributes, and if you use the systems biology tools and analyze the microbial behavior at community level, okay, so how they behave in the community level with different type of stress, you can also think about the metabolic engineering aspect of that particular microbe. And also if you use the system biology and ME both together, SBME all together, you can have more purposeful research to understand specific bioremediation and targeted bioremediation. I say targeted bioremediation. You want to degrade the mercury, you want to degrade pesticide, you want to degrade plastics, and if you see, there are a lot of things happening, and plastic pollution is a big problem, you know that. And if you see everywhere plastic, and those who are from Delhi, they know that there are so many dump sites. Okay, there is a big hill also. क्या नाम है उस हिल का? जो Balaswa. I know, because we pass through that. You know that bigger hill. You have seen that hill, dump side hill. How many of you have seen it? सबने देखा होगा, वो पूरा पहाड़ है. All पहाड़ means hill. Okay, so hill with full of all type of waste, and that hill is growing. Okay, because the dump site is growing, and nearby there are a lot of living people nearby. So the Rohini sector 16, 18, all I can remember. So whenever you pass by this highway and through that ring road, you will see that. So we are really on a very serious risk, and these type of techniques can be employed at a larger scale if the group is working on diversity of microbes and they have really a good consortia. I am not telling we can just degrade all the plastics and all that. Degrading plastic is a big challenge. So plastic is everywhere, and nowadays the risk is microplastic. You were talking about the neuro related, so yesterday night I read few papers by curiosity that how we can connect. I will share with you few papers where you can learn that. I was just also going through one paper where the effect of this endocrine disruptors, especially in the neurological disorders, endocrine disruptors, that is also a pollutant. Whatever shampoo you use, soap you use, okay, and various type of beauty products you use, lot of things are there. So that's why you use whatever you use, you don't know, and post use problems we came to know after some time. So these aspects can be covered, can be studied with the help of these tools. I will show you some tools and some tutorial materials in the coming. If you see this one, I am giving you some ideas for your research, so you can take these ideas as you wish. So if you see microbial communities, how they are in the polluted site, you use these tools and try to get the synthetic microbial consortia, SynCom. You read one paper which we published recently, that is a SynCom, synthetic microbial consortia, and use this synthetic microbial consortia for targeted bioremediation. If you want to have a targeted bioremediation, you can use this tool. Then you can read this tutorial material, that is the futuristic avenues of metabolic engineering techniques in bioremediation. Here only the techniques which are metabolic engineering based are given, and you can read it. This is a tutorial material. And now coming to the systems biology approaches. When we talk about synthetic microbial consortia, this one, okay, how to design this is a big challenge. So what are to be targeted? So if you want to design a synthetic microbial consortia in your own research, forget about bioremediation, in your own research, if you are working on gut microbiome, if you are working on gut brain axis, if you are working on various aspects of plant based plant growth promoting microorganisms, if you are working on some other area of cell culture, okay, so how to build up all those specific synthetic microbial consortia in that scale, or if you want to use synthetic biology in that aspect. So what will be the examples and what tools you can use has to be discussed here. So gene and pathway identification can be done through genomic tools. So it involves the sequencing of microbial genome. What you will do? You will do the sequencing of microbial genome, or sequencing of your plant genome, or sequencing of whatever work you are doing, and try to identify the catabolic gene and pathways involved in bioremediation, like we have done it. And then इसको फैन बंद करो, बहुत ठीक आएगी. Sneezing, then microbe dispersal. Once I sneeze, I will disperse a lot of microbes. Okay, so there are a lot of microbes here also, you know it. So diversity now, from Hyderabad, from Delhi, from Punjab. Okay, oh, Punjab wale students aaye, they came. They are not here today. They are absent. Okay, so we use the combination of the technologies and also these technology to assemble the genome assembly. That is also a new technique. So we are in next coming slide, we will discuss only the techniques part. Okay, and genome assembly, I think, is a very major challenge, and many people, they are using bigger systems. So for that you need a bigger system for genome assembly. So genome assembly, and also try to analyze the structure and function of specific genes. And if you use the NGS technologies, you can identify, monitor, you can also see the microbial communities which are involved in various aspects, and we are talking about the bioremediation here, so we will show you that one. Now, how you understand the microbial community using transcriptomics. What are the tools available and how these tools are to be used. So if you see, there are different type of pollutants. Those who are working on microbial bioremediation, they are working on remediation, they see there is diversity of pollutants. So how these techniques, especially the microbial community understanding and using transcriptomic tools, can be helpful. So you try to see that which is the specific microbial cell which is adapted to that particular pollutant, stress, under stress condition. Like if you go to some landfill site, you isolate microorganism, the microorganisms are living there in that condition only. It means they are adapted to that condition. They are still doing their job. Now you want to study that how they are doing the job, and can the remediation or that particular job can be initiated in a better way. In the same way, like probiotic is doing their job. So what is the need of more probiotic? They are doing the job in the body. Okay, so in the same way, whatever application you are doing, so here you try to see that if you want to design a consortia, you should have similar type of powerful microbial load, microbial population, and then mix them to become a consortium. So all these powerful microbes which are doing their job excellently in stress condition, if they are targeted towards specific type of metabolites or microbes for the remediation, they can definitely be thought about the consortia development. You cannot make a consortia of microbes which are not known for their work. Okay, you make a consortia. It is same like that. You see the cricket match yesterday. Okay, you make a team where you have an all-rounder. You should have a bowler. You should have a batter. You should have a wicket keeper, like that. And if you have more all-rounders, that is good. So we are talking about all-rounders. So in all the population, this is a diversity population, and identifying all-rounder is not easy. You have to give them bowling, you have to give them batting, you have to give them fielding, then try to understand that, okay, this is all-rounder, take it in the team. And the good team means good consortia will give you the match, give you the results. So in the same way here also, any area, like I think there are many people working on the area of specific related to the biotechnology innovations, especially the cell culture, okay, cancer biology and other tools which are used. So how they are able to generate various type of genomics data, how they are able to know that how these various type of mechanisms are happening in the body, what is the progression of the disease. So many aspects are there. So in the same way here, you try to see the consortia, you make a consortia of powerful organisms which you have studied and which you are knowing that they will do the job in the site. So polluted sites and their proteome analysis you do, and try to see that how they are able to, which type of protein is active. One gene, one enzyme theory, you might have heard about it, Biddle and Tatum. So the same story comes here. The theory is not changed. One enzyme, one gene, one enzyme theory. Okay. So which gene is targeting which enzyme and how the job of bioremediation is effective. So what specific gene is there, I will show you in the coming slide. And the microbial cell also secrete secondary metabolites, you know that, in response to various changes, and these metabolites can be analyzed through NMR, GCMS, LCMS and also HRMS. So this is another clue that how this is happening because of certain secondary metabolites also. So you can think about the secondary metabolites. Those who are working on plant system, they might be knowing about VOC, plant volatile materials, you know. So there is a term called MVOC, microbial plant metabolites, volatiles. They are also having very good significance. And those who are working on medicinal plants, they know that how these volatiles are quite important. So we talk about the tools. So you can use these tools to identify the metabolites. What are the metabolites? And there are techniques here. And there are some in silico tools which can be used to understand that. And these tools can help you to analyze the huge data available. And also they can helpful for giving the information about what cellular mechanism is happening, including the cellular fluxes. We are not taking up any class for or tutorial for flux. Okay, so probably we will have one tutorial materials flux, FBA. It is not covered here, but there is a separate thing that is called flux analysis, metabolic flux analysis. Okay, so this flux analysis gives you the importance about the physiological processes and how they are connected all together in a certain manner to give the outcome. So this flux can be studied, and also the pathway can be studied, that which pathway is important for the bioremediation attributes. So this approach will give you all intracellular and extracellular biochemical reactions and also enzyme pollutant reactions. So what enzyme is targeting what type of pollutant can be taken up. Now other tools you people are using, so let's discuss that also, like molecular docking and simulations. So I have given one example of bioremediation in the slide. We cannot give all the examples here. So taking bioremediation as a whole, I am taking up all, but you can try these tools. So molecular docking and simulation, everybody is using it. So these tools are also very quick. they tell you some a specific idea about what type of proteins are there, what enzymes are there, what are the substrates, how these are can be developed in a better way, and also the prediction. Okay, like if you are working on enzyme, so enzyme substrate prediction, which enzyme, and improving the capability of that enzyme or using the capability of this molecular docking and simulation. Then also trying to design the synthetic microbial consortia, and also the computational models, algorithm and other tools can be used. Now you see there are tools which we have given here, and this tool is based on means these tools can be used, and this is a tutorial material review paper which we published in 2020. Here you use all these tools and try to understand the modeling, virtual screening of the drugs. Those who are working on drug designing, they can also do that, drug designing, and you use molecular docking, you do comparative sequence analysis, you do pathway reconstruction, okay, to understand that how it is happening. And these are the tools: KEGG, EVAG, then BioRad base, Aroma, DEG, Bionomo. So many tools are there which are used, and you see these are the databases, okay, where you get the information about the, I am telling about the pollutants, okay, because this study we have done in our lab, so we use these databases to understand. Like if you want to know what pollutant you want to target, you have to see these databases, and these databases will give you the enzyme which is involved in these pollutant degradation. So you have this Aroma, DEG, Meta-Router, Binase, okay. Now there are certain limitations. In silico work has lot of limitations. So lot of limitation means lot of challenges. Limitations comes with challenges, and challenges comes with solution. So we should have known the limitations, we should have known the drawbacks. There are a lot of drawback in the in silico work, I am telling you. And in silico work, data management, too much data we get. That's why I was telling you, I could not complete yesterday the research manuscript part, but there are so much data available, and those data, how to compile all the data put together is a big challenge. So this limitation will not be only the data, this is a complexity and in the preparation of the samples, data handling, high cost, and also only in silico is not widely accepted. So only in silico work is not accepted. If you are able to tell us that, okay, I have a synthetic microbial consortia, it will degrade mercury. I have synthetic microbial consortia which will degrade the plastics. Nobody is going to believe it, because you need to prove it in the lab condition or in the field condition. Or you tell that, okay, I have a very good microbe which will give very good crop improvement, which will give the drought resistance crop development. So the rice plant will be able to. So it is all hypothetical, computational, it needs validations. Validation can be done only through wet lab. So validation is a very important component, and researchers are focusing to improve these techniques and overcome these limitations. So how to improve these techniques means combine two techniques together, because one technique is not sufficient. I told you, if you do modeling and simulation, you know crucial points, you get crucial lesions, or you also understand that, okay, these are the substrate. But if you do enzyme engineering, there are other aspects, like you do the kinetics, you do the growth curve, you do the time course, you understand the thermostability, so many aspects. All that is to be done by wet lab. So in the same way, you need to improve these techniques, make combinatory use, learn from it, and also try to see its validation through wet lab. Then there is a requirement to develop better annotation tools for the analysis of genes, metabolic pathway, metabolites involved in the degradation of pollutants. This is a big challenge, and this challenge I face almost every year, or I say. I told you that there is lot of data available. Lot of data matlab you have given the samples to some NGS people and they have given you all the data. How to analyze it? We got one data analysis in August. Still we are not able to analyze it. We got one data in 24. We were able to publish this year. 24 analysis was done. 23 analysis was done. But we were able to publish because we are not able to understand all the data together. And what is important? Excellent diagrams, excellent figures, excellent legends, but it is not significant. So you need to see that which is the pathway. Like I will give you very simple example just to avoid your confusion. There is a microbe which is able to degrade certain type of pollutant. You studied that microbe, you study the growth profile, you study its consortia, profiling and all that you have done. You mixed it with 2-3 microbes. But you got the data which is from all 3 profiles, like microbial alone, then consortia. Now you know that which is the pathway which is involved. So you have to see that pathway engineering tools, you have to see which pathway, which gene is giving the proper signals for bioremediation. So again you go for the analysis of this data and try to see the metabolic pathways involved. Then once you study the metabolic pathway, you don't know which type of flux is generating this pathway. So again you go through the flux part, and then which flux is controlled by which set of gene network. So this is a whole story which takes lot of time and it is incomplete, always incomplete. The story is not complete. So this is a big challenge, and again I will say you that if you want to study a microbial population, it is challenging. If you study a microbe, it is also challenging. If you study any single application, that is also challenging. So everything comes with using the tools, but when you use 2-3 tools, you understand to a little bit. Means you go certain beyond that, like you enter in a building. First time when you enter, you see lot of rooms are there. Second time you enter, you see more rooms there, and sometimes you don't want to go that side, you go there. So it is like that. You have to explore the whole building to understand which side, what components are there, how many people are living in the building. Okay, if you go through this building, how many teachers are sitting, where MSc classes are there, where practical is happening. If you don't explore, you will never learn. So you will never reach to that point. So I will show you in the map ki this is a PG lab, this is UG lab, this is research lab, this I can show you everything here. I can tell you, but once you explore, in the same way, analysis of gene, annotation of gene is a very challenging aspect. Very challenging aspect. So we need to think about those aspects and try to see that how these analysis can be done. And if you see, this is a good review, designing synthetic microbial communities for effectual bioremediation. Why we are trying to design the synthetic microbial community is discussed here, why it is important. And please read this review. This is also very well cited. I think gene network I will take up after the break, because Shrabni, we need to break at 11, right? So we'll break now and then take up the gene network. It will not take much time, and then the tutorial will be run later on. So we take a break. My major concern is learning through combinatory techniques. What I feel during my last 24 years experience is that the major challenge is learning new techniques, adapting new techniques to your research. The biggest challenge, because we are always having a safeguard, okay, we don't want to go that side, okay. So those places where we want to avoid ourselves, if we are able to learn some of them, we can be comfortable and we can learn some other things. Like Mohit did excellent work in his research during his PhD, but he has not analyzed yet metabolic network. He has not yet analyzed the pathways. So if he targets all those aspects, he can do better. In the same way, his students and many students, like in our group, in your specific area, in your senior PhD students, they are working on one area and they finish completing that area without touching other areas. So this course will give you some clues, some software. I cannot say that you use all the software, all techniques, everything, but I have shown you the successful examples. I will show you that also. And if you read the papers which are published by our group, they are based on multi techniques use. They are not using one technique. They are using at least 2 or 3 techniques. If you use one technique, you will understand little bit. But you use multi-omics techniques, multi-techniques, and interdisciplinary evolution of the technique is important. So we are afraid of kinetic models. You see that kinetic models कौन बनाता है? बहुत मुश्किल है. Mathematics है. You know, I have discussed with our mathematics friends here. Okay, and one mathematics professor was there here in the inaugural function. He was sitting here. So that's why people ask me how you are connected to that. I am learning from him to understand how to avoid the my effort for mathematics. So we are afraid about mathematics. कैसे करेंगे? Calculation कैसे होगा? So if you are having a good understanding with mathematics people, they can teach you, and that can be easy for them, which is very difficult. Okay. So you should try to see that. And in the same way, computational biology aspect. So Professor Azad is not a biology person. He is a physics, mathematics person. So he is a better person to teach us. Everything he can teach us in a better way. And my students are learning from him. Last, I think, 2 years they are learning. So earlier also some students they learn, but it depends on your learning capability. You get lot of networking, you think मैं ये कर लूँगी, कर लूँगा, ये कर लूँगा, but it is not happening until unless you really think about interdisciplinary involvement. Okay, so with this